everybody. All right, so this is the last video on the digestive system, which is an accessory digestive organ called the liver. Can't live without a liver. The liver has so many functions.
There are 4 sections I will be discussing on this video. First is metabolic regulation. Second is hematological regulation. Third is production and functions of bile. And fourth are the models. If you want to skip over the functions of the liver, just go straight to the section on models. Metabolic regulation. The liver is the primary organ which regulates the composition of blood. Blood from the digestive tract enters the liver. Hepatocytes, or liver cells, extract those nutrients and toxins from the blood for removal and storage. Finally, the liver mobilizes reserves or performs synthetic activities to correct nutrient deficiencies. First, there's carbohydrate metabolism. The liver monitors blood glucose levels by using hormones such as glucagon and insulin. Second, there's lipid metabolism. When lipid levels are high in the blood, the liver stores them. When lipid levels are low in the blood, the liver breaks down its storage. Now, this only happens when lipid levels are high because the lipids bypass the hepatic portal circulation. Third, there's amino acid metabolism. The liver can store the amino acids or synthesize proteins from those amino acids. Fourth, there's waste removal. When converting amino acids to lipids or carbohydrates, an amino group is stripped off in a process called deamination. As a result, ammonia, a harmful toxic product, is formed. The liver converts ammonia to a harmless compound called urea. Urea is later on excreted by the kidneys. The liver removes other waste products, circulating toxins, and drugs from the blood. Fifth, there's vitamin storage. Fat-soluble vitamins and vitamin B12 are stored in the liver. Sixth, there's mineral storage. The liver converts iron reserves to ferritin. Finally, there's drug inactivation, number 7. The liver removes and breaks down circulating drugs, limiting the duration of their effects. Hematological regulation. The liver receives approximately 25% of the cardiac output. The liver is the largest blood reservoir in the body. The hematological functions of the liver are as follows. First, there is phagocytosis and antigen presentation. Stellate macrophages located in the liver sinusoids engulf old or damaged red blood cells, cellular debris, and pathogens, removing them from the bloodstream. There are also antigen-presenting cells that can stimulate an immune response. Second, there is synthesis of plasma proteins. Hepatocytes synthesize and release most of the plasma proteins, including albumins, clotting proteins, and complement proteins. Third, removal of circulating hormones. The liver is the primary site for the absorption and recycling of epinephrine, norepinephrine, and other hormones, such as sex hormones and corticosteroids. The liver also absorbs vitamin D3. The liver converts vitamin D3 into an intermediary product, which is later on returned to the blood. The kidney absorbs the intermediary to generate calcitriol, a hormone important to calcium. Fourth is removal of antibodies. The liver absorbs and breaks down antibodies, releasing amino acids for recycling. Fifth, removal or storage of toxins. The liver absorbs lipid-soluble toxins in a diet, such as DDT, and stores them in lipid deposits where they do not disrupt cellular functions. The liver removes other toxins from the blood and either breaks them down or excretes them in the bile. Production and functions of bile. After the liver synthesizes bile, the bile travels via a series of ducts through an ampulla and into the lumen of the duodenum. Bile consists mostly of water, small amounts of ions, bilirubin, cholesterol, and several lipids known as bile salts. Bile salts aid in digestion of lipids. Most dietary lipids are not water soluble. The larger the droplet, the more lipids are inside, isolated from lipid digestive enzymes like pancreatic lipase. Bile salts break apart these lipid droplets in a process called emulsification. Emulsification creates tiny little droplets with a superficial coating of bile salts. The layer of bile salts facilitates the interaction between the lipids and lipid digestive enzymes from the pancreas. When lipid digestion is complete, bile salts facilitate the absorption of these emulsification droplets by the intestinal epithelium. More than 90% of the bile salts are absorbed by the intestinal epithelium of the ileum. The reabsorbed bile salts enter the hepatic portal circulation. The liver then collects and recycles the bile salts. The cycling of bile salts from the liver to the intestines back to the liver is called the enterohepatic circulation of bile.
So anyways, the liver, very important. So let's look at what a liver looks like from the outside. This is a liver here.
Anatomical models.
Okay, this is the bigger lobe on the right, so this is the right lobe. Smaller lobe is the left lobe. Okay, we have two other lobes here. This one here looks kind of like a square. This is called a quadrate. Okay, and this one's called a caudate. Okay, caudal, think of like tail, caudal, just longer. So caudate, quadrate, left lobe, right lobe. Here we have the falciform ligament. Kind of just holds the liver firm anteriorly. Okay. And I believe this one back here is the round ligament. Up here, I believe, is the coronary ligament. Okay. I want to see what it looks like in the inside. We have this model here. Okay, a bunch of veins. Okay, now there's two different color of veins in here. See if you can see. Okay, so the purplish one here, okay, here you can see the triad. This will be the portal vein, okay? So this portal vein is going to bring those nutrients, so that's why it's kind of like a purple color. There's two veins in here. There's two veins. One is more purple and the other one's more blue. Okay, this is more blue, more purple. So the portal vein is going to bring this venous blood, brings those nutrients into the liver, drops it off, and then the hepatocytes metabolize it. Then the remaining blood will continue via the hepatic veins, and those hepatic veins eventually will connect to this guy here called the inferior vena cava. Oh, here you can see the hepatic veins in the back, hepatic veins, and then here you have the inferior vena cava. Inferior vena cava. Okay, so that's what it looks like in the inside. A lot of blood going inside this thing. Blood with nutrients, and then blood with just poor oxygen, which eventually is going to go to the lungs and get oxygenated. Okay, so let's look at this diagram here. So you see this hexagonal shaped structure? Okay, hexagonal shaped structure. Okay, that's called a liver lobule. Liver lobule, and there's 100,000 of those liver lobules in our liver. And the liver lobule is going to be composed of several structures.
First is the portal triad.
Those three structures right there, portal triad. Interlobular artery, interlobular vein, and interlobular bile duct. And that triad is in the corner of each liver lobule, because it's a hexagonal shaped structure, so it's in each corner. And now in the center here, we have a central vein. A central vein. So the nutrients are going to go from the interlobular vein, go to the sinusoids, which are liver capillaries. Sinusoids will drop off those nutrients to the hepatocytes. And whatever's left over in the blood is going to go to the central vein and eventually up to the hepatic vein, inferior vena cava, and in the 14 steps of the heart. All right, so let's look at this, the heaviest model I have in this lab, except for the box of bones, but well, this is pretty close. Okay, so anyways, this is a cross-section of a liver lobule. Remember, there's 100,000 of these liver lobules. All right, so here's their triad here. This is a lymphatic vessel, so this is not part of the triad. So you should start learning the new way with the new book that we just got. We're in the 11th edition already, but you just say interlobulars. These are all interlobulars. The interlobular artery, interlobular vein, and interlobular bile duct. Okay. Here's the central vein. Here's the liver sinusoids. Okay. So basically you're going to have these triad, you're going to have 6 of these going around. That. One here, one here, one here, one there, and there. 6 going around the central vein. But all these portal veins are going to take that blood to the liver sinusoids into the central vein. All 6 of them. They're all going to take the blood into the central vein via the liver sinusoids, which are these. Then the liver sinusoids, which are the liver capillaries, are going to have these structures in it. Let me see if I can see it here. These things right here, these are called the Kupffer cells. And the Kupffer cells, they're going to monitor the filtrate as it's going from the liver sinusoids into the hepatocytes. Okay, so let's look at some other components. Okay, these are called hepatocytes or liver cells. Okay, hepatocytes or liver cells. So one of the functions that they do is to produce bile. Okay, so the bile has bile salts that break down fat. The bile is going to eventually go to these little smaller lymphatic capillaries called bile canaliculi. Okay, so the bile canaliculi can take that bile and go to the interlobular bile duct. And the interlobular bile ducts, all 600,000, want to come together and eventually form the left and right hepatic ducts. Okay, this is a piece of the liver, by the way. This is the gallbladder. This goes to this model. It's broken off here, but the idea connects like that. So the left and right hepatic duct takes that bile duct and goes to the common hepatic duct. Now the common hepatic duct joins the cystic duct that comes from the gallbladder. And the gallbladder, what it does is that it gets that bile and makes it super concentrated. Okay. So the common hepatic duct joins the cystic duct to form the common bile duct. Okay. And then the common bile duct will come down. This common bile duct will come down and come down here, and the common bile duct will join the pancreatic duct to form the ampulla vater, which is surrounded by the sphincter of Oddi, and it's regulated by the hormone CCK. Okay, so that'll allow bile to go from the liver to the gallbladder to get concentrated, down the common bile duct, into the duodenum to break down that fat. Okay. So yeah, that's one of the functions of the liver, is that it produces bile to break down fat, among many other things. Okay. The last subject on the test, the last test is going to be reproductive and urinary, so make sure to stay tuned for that, or for digestive system. That's it. All right, guys, take care. Like and subscribe. Talk to you later. Bye.